Here you can see all of the nucleotides and nucleic acids that we are looking at are built up on 5-membered aldehyde sugar, a pentose, that either has 2 OH groups here, which look like this. They're both on the right-hand side. This is called ribofuranose and its long name, or in its common name, ribose. So ribose is used as a sugar. Again, it is a furanose type sugar. Now, the other type of sugar that is often used, and we see it in DNA instead of ribose, which is used in RNA, that's where the R comes from, is deoxyribofuranes, or deoxyribose is its common name that you'll see. The D in deoxy gives us our DNA. Deoxyribose is instead missing that first OH group. You can see, in fact, in our first example here, this sugar that is highlighted in pink, this OH group that is highlighted in pink is the OH group that is versatile. So depending on if an OH group is there or it is not, you either have ribose, which is all right, remember, all on the right-hand side OH groups, or when missing this OH group, you have deoxyribose. Now, the sugar itself, the pentose sugar base, also has a formation depending on, remember, where this glycosidic bond is pointing. In a nucleotide formation, our nucleotide makes a bond here on the glycosidic carbon. And remember that depending on the positioning of this group, either same side as our CH2OH group or opposite side of our CH2OH group, this can be called beta or alpha. And note here that the beta form of the sugar is the one that is preferenced in both RNA and DNA.